News release. Thursday, June 22, 202-3. National Institute of Health-supported trial first step in developing new approach to treat myasthenia gravis. Evidence from a small-scale clinical trial suggests that a variation of the advanced blood cancer immunotherapy known as CAR-T could be adapted to treat myasthenia gravis, an autoimmune disorder of the nervous system. The modified CAR-T therapy, short for chimeric antigen receptor T cell, used by scientists offers the potential for a longer-lasting reduction in myasthenia gravis symptoms and was well tolerated without significant adverse effects. The study, published in The Lancet Neurology, was supported by a small business grant from the National Institute of Neurological Disorders and Stroke (NINDS), a part of the National Institutes of Health, and sponsored by Cartesian Therapeutics, Gaithersburg, Maryland. Repurposing a groundbreaking therapy such as CAR-T to potentially treat a neurological disorder shows the versatility of immunotherapies in instances where there are limited to no treatment options, said Emily Caporello, Ph.D., director of the NINDS Small Business Program. Myasthenia gravis is a chronic autoimmune disorder most often caused when the body's immune system attacks a protein found where nerve cells communicate with muscles. The disease is marked by muscle weakness that worsens after periods of activity and can improve somewhat after rest. Current treatments focus on controlling symptoms, primarily muscle weakness. In the study, 14 people with generalized myasthenia gravis received varying doses of a modified form of CAR-T therapy known as discartesminus-8, targeting the cells responsible for producing myasthenia gravis-causing antibodies. The ideal dosage was determined to be once weekly for 6 weeks. Early data on the effectiveness of the treatment are promising, but additional clinical studies are needed to evaluate the therapy's efficacy. 3 patients receiving discartesminus-8 showed complete or near complete elimination of their symptoms, which continued 6 months following treatment. 2 others no longer required chronic intravenous immunoglobulin treatment, which is currently used in some severe MG cases. What we saw were deep, durable responses to discartesminus-8 that persisted for at least 6 months following treatment, said Murad V. Kalioglu, MD, Ph.D., President and CEO of Cartesian Therapeutics. We have now begun a larger randomized, placebo-controlled study, which is the first of its kind for an engineered adoptive cell therapy. CAR-T therapy involves taking a patient's T cells, a key part of the immune system that can recognize and destroy invading pathogens, and reprogramming them to attack a specific target. In the case of blood cancers, the new target is the cancer itself. For myasthenia gravis, the target is the corrupted cells that make the damaging antibodies. Many immunotherapies, including CAR-T, carry the risk of serious side effects that, while tolerable in the cases of advanced cancer, prohibit its use in more chronic conditions such as myasthenia gravis. Normally, T cells are reprogrammed with DNA, which persists within the cells and is copied every time the cells divide. This can lead to an amplified effect and serious side effects. To avoid this side effect, discartesminus-8 uses messenger RNA mRNA, which does not duplicate when cells divide, instead of DNA to reprogram T cells. The result is a short course of treatment that is given multiple times instead of the single-dose regimen normally used in DNA-programmed CAR-T therapy. The primary aim of this trial was to determine the ideal dosing for discartesminus-8 that effectively reduces muscle weakness symptoms with minimal side effects. Discartesminus-8 therapy is now being tested in a larger clinical trial to determine its ability to reduce myasthenia gravis symptoms. Importantly, this trial will also include a placebo group, which is an important control to confirm that any observed improvement is due to the treatment rather than any unrelated effects. This trial was supported by a grant from the INDS Small Business Program and SON 115,426-1 which is designed to encourage research and development that can lead to commercial therapeutic products. The clinical trial reported in this study and the ongoing Phase 2 trial are registered on clinicaltrials.gov NC T04146051. About National Institute of Neurological Disorders and Stroke NINDS is the nation's leading funder of research on the brain and nervous system. The mission of NINDS is to seek fundamental knowledge about the brain and nervous system and to use that knowledge to reduce the burden of neurological disease. About the National Institutes of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health Turning Discovery into Health Registered. Granite v. Benetter M. Kurtoglu M. et al. Safety and clinical activity of autologous RNA chimeric antigen receptor T-cell therapy in myasthenia gravis MG-1, a prospective, multicenter, open-label, non-randomized Phase 1/2 study. Lancet Neural, June 21, 202-3. DOI 10.1016/PIIS 1474-4422-394-1. This page last reviewed on June 22, 202-3.